Hello and welcome to BioRules video lessons. Today we are going to be talking about indicator species. So indicator species are species that indicate environmental quality. So sometimes you have a disturbance in an environment. This disturbance may cause health issues to the population. Okay, they may indicate environmental quality, how that environment is changed or damaged. Okay, it shows the deterioration and usually is strongly related to pollution. Pollution is one big environmental disturbance. And there are different ways for us to see pollution. For air pollution, the lichens are very good indicator species. So if we know the lichens that are in our place, we can make quite a picture of how much pollution, air pollution, is in that place. Okay, and for water pollution, good indicators are invertebrates, like the larvae of aquatic insects. So they will indicate the quality of the water, because they may be sensitive or not for water pollution. So let's start talking about the lichens. They indicate air quality because they may be or may not be sensitive to some sulfides and some nitrous gases. So if you have much of the sulfides or nitrous gases, some species are going to thrive, other species are not going to be able to thrive or to survive at all. So just to give you an example here, you have one species, this Usnea, in the United Kingdom. On the left, we have green areas that you have more pollution, the yellow less pollution, and the orange even less pollution. And on the right, we have the distribution of this lichen. Apparently, this lichen is not very fond of highly polluted sulfide gases. So where you have strong sulfides, you have, for example, in this area here is very clear. This area here is very clear here that we have quite a strong pollution because of the concentration of the lichens. Another cool example, here is not indicating the pollution, but the lichens biodiversity, so going from 3.3 to 5.5, is correlated to lung cancer mortality in some cities. So is lichen cause lung cancer? No, probably what's happening here is that lung cancer is related to the air quality. Lichens biodiversity, we know, is related to air quality. So what is happening is that the lichens here are actually indicating the quality of the air, and the quality of the air is having an impact on the mortality by lung cancer. Indicator species, there it is. Okay. Interesting about the indicator species that it's quite easy to be used with a simple table with pictures of lichens from a certain region. Now you can have the ones that are from highly polluted areas, moderately polluted, slightly polluted, minimal or no pollution. So if you find those guys over there, you can have an idea of how much pollution you're having in the place. You don't need fancy equipment. You don't need expensive stuff. You don't need specialized technicians. No, a reasonably educated person with a table like this can already qualify and maybe even quantify pollution in a certain place. Okay? So let's talk about water now. Water microinvertebrates, they are very good to talk about water pollution because these insects may be sensitive or not to pollution. Mayflies, for example, they are sensitive to polluted water, and they are quite easy to identify. The adults are on the left, the youngs that live in the water are on the right. So basically, if you have a little table like this, now if I give it a close here, we have clean water organisms. So you have those that are the hydra, mollusk, limnia, the larva of damselfly, freshwater shrimp. So those are the clean water, okay? And then you have the slightly polluted and polluted. Once you have a table like this one here, it's quite easy for you to qualify the water pollution or maybe even quantify the water pollution. You see? A little table like that, group 1, group 2, 3, and 4, for the ones that are intolerant to pollution over here, and for the ones that are tolerant to pollution over there. So, indicator species. Water invertebrates, they are good indicators. Okay? They have a wide range of tolerance. Some are very sensitive to pollution. Some are very tolerant to pollution. Okay? They are easy to identify, at least to the level that is necessary for indicator species work. They are quite easy to identify. Okay? If you have an ID guide, that's easy to build, easy to use, even non-trained volunteers can use them. So it's very easy, simple, and cheap to be using this. Okay? So to illustrate how you can quantify, not only qualify pollution, there is one thing called biotic index. The biotic index is trying to give a number to the sensitivity of a species in a certain area. So the formula for the biotic index is this, where the big N is the total number of individuals that we collect in an area. The small n is the number of individuals for each species, and the A is the tolerance rating for a certain species. I'll give you an example here. So the A here for the tubifax worm is 1. Why? Because it's highly sensitive, so we give a number of 1. We give a low number for the ones that are more tolerant. And the stonefly nymph, that's very sensitive, has a low tolerance for pollution, we give a high number. In this way, when you calculate the biotic index, if you have a big number, that means that place is quite clean. Low pollution. If you have a low number, then this place is going to be quite polluted. So to exercise this, I'll give you an example, and I'll give you some time to calculate. So there is a sample that we have in a certain place. We found 8 stonefly nymphs, we found 1 freshwater shrimp, and we found 1 tubifax worm. So calculate the biotic index for this place. I'll give you some time, pause the video. When you finish, carry on, and we are going to check to see if you got the same value as I did here. Okay, so how do we do that? 8 stoneflies times 9 of its tolerance. 1 freshwater shrimp times 5 of its tolerance. And 1 tubifax worm times 1 of its tolerance. So when we do that, we are going to be finding a certain number, divide by 10, because you have 8 plus 1 plus 1, there are 10 individuals. So this will be 78 divided by 10. This gives us a tolerance of 7.8. Another example, 4 stonefly nymphs, 10 freshwater shrimps, and 6 tubifax. The same species, no change in the richness, the same species, but just with different numbers. Pause the video to calculate this here and come back when you finish. Okay, let's see if you have calculated it correctly. So you have 4 stonefly nymphs times 9, 10 freshwater shrimps times 5, and 6 tubifax times 1. And we have a total of 20 individuals. So this will give us a total of 4.6. This is the biotic index of site number 2. What this means? Site number 1, 7.8 is cleaner. Site number 2, 4.6 is more polluted. This is how we use the biotic index. So you can quantify by the tolerance of a certain individual in the place the level of pollution or any other disturbance that you have in that area. Remembering that the tolerance level is going to be smaller for the ones that are more tolerant to high pollution, and it's bigger for the ones that they are more sensitive. We want our result to be the bigger, the better. Okay? So now we have here a little questionnaire with some questions for you to see if you learned well this lesson. I hope you had fun, and I'll see you in the next class.